So welcome to my presentation. My name's Tim. I'll be presenting my preliminary results from my master's thesis so far, where I've taken kind of a big switch from being mostly into the field and then doing now a little bit of data sleuthing. And so as previous people have mentioned before, our freshwater fish communities are facing numerous numbers of stressors. And so I was motivated for my thesis to understand how we can predict fish communities under these future conditions. So under our current prediction frameworks, if we're trying to figure out what fish from the region are going to be found at a local lake, we're going to first consider the lake's abiotic conditions. And so in our example here, largemouth bass are going to be excluded. Next, we're going to consider what biotic interactions might occur. And so in this example, northern pike are going to exclude our brown trout. And so we're going to end up with a lake with just pike in it. But could there be a situation where both of these species can coexist in the same lake together? And so that may happen if we consider an interaction between environment and biotic interactions. And so this abiotic and biotic relationship is exactly what this group looked at in 2014, where they found that in lakes with lower temperatures, both brown trout and pike were able to coexist with each other. And they found that in lakes with higher temperatures, northern pike exhibited higher predation pressure on brown trout and they were excluded. And so this kind of relationship between the biotic interactions and the environment can be considered as context dependency, where the importance and magnitude of a mechanism depends on abiotic and biotic factors. And so for my project, we were looking at how this context dependency might affect our ability to predict fish communities into the future. And so this is a really big question to tackle, so we broke it down into two smaller parts. Our first question was, are certain mechanisms like competition or predation more sensitive to context dependency than others? And secondly, does this context dependency scale up to drive our regional level diversity patterns? And so to answer these questions, we used a large data set of lake communities in Ontario. From there, we inferred mechanisms. And so for our project, we were looking at positive and negative biotic interactions. And from there, we looked at how these mechanisms varied with the environment. So we're really grateful for our data set, which comes from the Ontario Broad Scale Monitoring Program. It's got a great scope. It's got over 40 species spread out over 700 lakes. We do have abundance data, but right now we're just using the presence absence data. And it also comes with an incredible wealth of environmental variables. We have 105 of them, and so that's pretty overwhelming. We split them up into 6 relevant categories and did a principal components analysis to make it more operational. And so this comes to the really exciting part where we try to infer mechanisms that might be shaping our communities, and we used co-occurrence analysis. So if you're not familiar with co-occurrence analysis, if we have a bunch of lakes here, and we have 2 species, in this example, triangles and circles, and they co-occur in the same lake together more often than not, we describe that pattern as positive co-occurrence. And that can be due to mechanisms like environmental filtering or biotic interactions like facilitation. If we have 2 species, circle and squares, which don't occur in the same lakes together, we describe that as negative co-occurrence, and that can be due to mechanisms like environmental filtering or biotic interactions like competition and predation. And last but not least, if we have a species like these pentagons here, which are spread out randomly, you guessed it, it's random co-occurrence, and that's due to random processes. And so we did some fun math here with Markov networks, where we can represent these patterns with links between species here. So triangle and circles is represented with a red link, which represents a positive co-occurrence pattern. And this also lets us explore how these relationships change across environmental gradients. And we also added a little environmental constraint null model, which lets us isolate these biotic interactions from environmental filtering, which can result in both positive and negative co-occurrence patterns. And so from kind of this big global model, we can do something really fun, which is estimate association networks for each lake in the data set. So here are 3 example association networks. They come from 3 different lakes, but each of these lakes have the same species in them, where the red lines represent possible positive associations and blue lines represent possible negative associations between species. And so I'm just going to highlight here's an example. This is a predicted association between northern pike and walleye. So you'll see in this lake here, they have a negative effect on each other. And on these 2 lakes, they have a positive effect on each other. And so this is exactly what our first question is trying to tackle. We're trying to understand if the environment is going to drive whether or not a species is more likely to be interacting with each other negatively or positively, and how that kind of comes together to shape the community. So we'll do some metrics here. So if we're trying to understand how some interactions might be shaping a community, we can use a network of heterogeneity, for example. So if we're calculating the heterogeneity metric for the negative interactions on a lake, if it has a score of 1, it means that one species, for example, it could be walleye, is having a negative interaction on all the other species in that lake. Versus if there's a score of 0, it could mean that all the species are having a negative interaction with each other. We can also do a metric of modularity, which kind of looks at how species are interacting with each other in big groups, small groups, are they clustered? And so these metrics can give you a really interesting idea of how these fish species might be interacting with each other to shape communities. And so we calculated these metrics of heterogeneity and modularity for all the negative interactions in each lake and for all the positive interactions in each lake as well. And then we can compare that with the environment. And so these are very preliminary results. So these are results for the negative network. So what this plot here is a redundancy analysis where we put in our explanatory variables, which are our environmental variables, and see how much the environment can explain these differences in our network metrics. And so this is for the negative interactions. So if the arrows are pointing in the same direction, they have a positive relationship with each other. If they're in the opposite direction, they have a negative relationship with each other. And so we see that a lot of these variables are positively associated with our negative metrics. And the environment can explain around 15% of our network structure. We did the same thing with our positive interactions, where you can see that a lot of the environmental variables have an opposite effect from the negative network on our network metrics. And here the environment actually explains just a little bit more about the changes in our network structure. And so just as a quick summary here, in terms of predictability, our positive interactions seem to be a little bit more sensitive to the environment, and that for both our positive and negative interactions, our network structure can be partly explained by our environmental variables. And so we have a lot of work to do still for this step here, including adding other metrics in, and also starting to interpret our environmental variables. For example, climate PC2 was an important variable, and that was related to hypolimnion depth and stratification. And like we saw in the previous presentation, stratification can have an important effect on how species interact with each other. So it's nice to see that kind of come out in our models. And leading up from that, our second question was whether or not these network metrics can kind of scale up and predict species diversity. And so these are all next steps. For diversity, there's always the standard method of looking at species richness, but there's another really cool way of looking at diversity where you can look at if a combination of species is rare or common. And so across Ontario, we can see that in the north, lakes tend to be more common and less unique in their composition compared to the south. And then we can look at how those diversity metrics compare with our metrics of network structure from our previous section. And we're also expanding it into some other metrics that I won't follow up with here. And so we're hoping that the lessons that we learned from answering these questions, so for example, looking at if negative interactions kind of change more the environment than positive interactions, can help inform how we can make predictions into the future under future climate situations. So for example, if we're predicting an expansion of smallmouth bass into the north, and we think that they might exclude certain prey species, we can use the lessons that we learned from this project to kind of better inform and be more realistic with our predictions. And so I just want to give a big shout out to the government of Ontario for giving our data sets, everyone in my lab, and also the AFS Quebec Student Subunit for telling me to come present here. And I'll be happy to take questions later.